Welcome back to The Explainer. Today, we're zooming way in, down to the microscopic warfare happening inside your body right at this very second. We're going to unpack the fascinating details from Chapter 360 and explore the incredible mechanisms that keep us alive, from the initial breach of our physical barriers to the deployment of some highly specialized cellular weaponry.
Here's our battle plan for today: the human defense strategy, first responders with innate immunity, the messengers and bridges, our special forces, the adaptive immunity, and finally, humoral versus cell-mediated defense. Let's get right into it.
Section 1. The Human Defense Strategy.
So, the big picture here is that we essentially have two main defensive armies. On one hand, we have the ancient, innate immune system. We actually inherited this from invertebrates. It's hardwired directly into our genetics, using germline-encoded proteins to instantly spot common danger patterns on pathogens. But on the other hand, we have the adaptive immune system. This one is exclusive to vertebrates. It evolved much more recently. It relies on T and B lymphocytes that actually rearrange their own genes to recognize highly specific, unique threats.
Now, for this dual army setup to actually work without, destroying our own bodies in the crossfire, it rests on three massive pillars. 1. It needs incredible diversity to recognize pretty much any threat imaginable. 2. Immune memory. It has to remember a past invader so it can fight it off way faster the next time. And 3, and this is a big one, immunologic tolerance. The system absolutely must be able to ignore our own healthy cells and harmless foreign molecules, like the good bacteria in our gut.
Section 2. First responders. Innate immunity.
When a pathogen breaches the perimeter, our first responders don't have days to figure out what they're up against. They rely on pattern recognition receptors, or PRRs. Think of protein families like the toll-like receptors. They act like molecular tripwires perfectly tuned to detect universal danger signals called PAMPs, or pathogen-associated molecular patterns. These are conserved structures that the pathogen absolutely needs to survive, like bacterial endotoxins, so the bugs can't just mutate them away to hide from our defenses.
A perfect example of one of these frontline defenders is the macrophage. When its receptors trip that alarm, it goes right on the offensive and physically engulfs the invading bacteria, which is a process we call phagocytosis. But it doesn't just eat the threat and call it a day. It immediately starts sounding a chemical alarm to rally more backup to the infection site.
Working right alongside those phagocytic cells is a brutal, soluble defense network known as the complement system. It's basically a relentless chain reaction of plasma enzymes. Whether it's triggered by the classic, MBL, or alternative pathways, the end result is always the same terminal pathway. These circulating proteins assemble into a literal membrane attack complex that punches holes right through the pathogen's cellular membrane. It just instantly bursts them apart.
Which brings us to Section 3, the messengers and bridges.
Those local defenders are fighting hard, but they need to call for backup. They coordinate this using an intricate chemical radio network made of cytokines. These are soluble proteins that regulate the growth and activation of other immune cells. At the same time, they drop a specific subtype of cytokines called chemokines. If cytokines are the radio call shouting, "We need help," chemokines are the GPS coordinates telling those reinforcements exactly where to go.
Amidst all this chemical crosstalk, you have the most crucial physical messengers, dendritic cells. These cells are the literal bridge between the early innate response and the heavy-hitting adaptive response. After they encounter a pathogen, they grab fragments of it, travel all the way to the draining lymph nodes, and present these foreign antigens directly to naive T lymphocytes. They're basically knocking on the general's door, officially requesting the special forces.
That takes us to Section 4, Special Forces, Adaptive Immunity.
Now, unlike the rigid split-second innate system, the adaptive system takes its time. It's a delayed response because specific lymphocytes have to activate and multiply. But the trade-off, because it uses gene-rearranged clonotypic receptors, it gives us practically infinite specificity to target the exact strain of an invader. Plus, it possesses immunologic memory. If that exact same pathogen ever tries its luck again, the adaptive response will crush it almost instantly.
The absolute rock stars of this adaptive army are the T and B lymphocytes. What's crazy is that even though they fight in completely different ways, both of these major cell lineages derive from the exact same hematopoietic stem cell. T cells go off to mature in the thymus, while B cells stick around and mature in the bone marrow. But together, they form the 1-2 punch of our adaptive immunity.
Let's break down how they fight in Section 5. Humoral versus cell-mediated defense.
B cells are the commanders of humoral immunity.
When a B cell recognizes its specific target, it morphs into a biological weapons factory, pumping out highly specific immunoglobulin molecules, antibodies, into our blood and mucosal fluids. These antibodies lock onto circulating microbes or toxins, totally neutralizing them, and tagging them so phagocytes can easily clear them out.
T cells, on the other hand, run our cell-mediated immunity. They aren't just tossing weapons into the bloodstream, they like to get up close and personal. Using their unique T cell receptors, or TCRs, they scan the surfaces of antigen-presenting cells, looking for specific pathogen fragments physically bound within MHC molecules. This lets cytotoxic T cells directly identify and destroy infected host cells, or it allows helper T cells to manage the broader immune response.
And the way helper T cells tailor the attack is just brilliant. Depending on the threat, they differentiate into specific subsets. Th1 cells deploy interferon gamma to fight intracellular microbes like viruses. Th2 cells release interleukins 4 and 5 to drive antibody production against parasites. Th17 cells handle extracellular bacteria and fungi. And we absolutely cannot forget regulatory T cells, or Tregs. They produce interleukin 10 to hit the brakes, modulating the response to prevent runaway inflammation once the battle's won.
So, bringing all this incredible biology together, a pathogen's journey inside us ends through 5 highly coordinated phases. It starts with leukocyte migration, escalates to innate nonspecific recognition, steps up to specific adaptive recognition, amplifies the inflammation to pull in reinforcements, and finally ends with total destruction and clearance of the antigen.
Now, why does knowing all this matter? Because it shows us exactly why that concept of tolerance we mentioned earlier is so vital. When this incredibly lethal, complex system loses its tolerance, maybe the regulatory T cells fail, or a barrier like the gut breaks down, it can start targeting our own normal flora or harmless food antigens like gluten. And that's exactly what drives devastating autoimmune and inflammatory conditions like Crohn's or celiac disease.
Knowing just how perfectly engineered this system is to remember and destroy, it leaves us with a pretty terrifying question. What happens when the immune system's exquisite memory and specialized special forces are permanently turned against the host's own healthy tissues? And how can modern medicine possibly intervene when our own microscopic army goes rogue? Definitely something to think about. Thanks for joining this explainer.